Picking up where we left off in 15-2, we're in chapter 15. We are looking at mRNA processing in eukaryotic cells for Bio 121. And what I want, again, to stress to you is that Mother Nature's figured out a way to transcribe a piece of information that will need to be manipulated so that we can actually begin to now translate whatever the information is in here, the final product, to make proteins. What's going to happen is our primary RNA transcript, we would probably call this pre-mRNA, is going to have a 5' methyl G cap slapped on it. You still have some 5' untranslated region here. But the interesting part is, look what happened to those introns. Those introns were cut out. We left on a 3' untranslated region, and we slapped on these polyadenylations, and these polyadenylations, as I said a moment ago, along with the 5' methyl G cap, allow for stability. So how do we get rid of those introns? We use a thing that's called a spliceosome, and what's going to happen is the GU and the AG here of the sequence in the intron itself allow for the spliceosome and small nuclear riboproteins to pull these structures together, and you now cut that out, leaving this GPA mark here, where you now have the exon 1 and exon 2 spliced together correctly in frame. Now, that is tricky. Okay? Obviously, mistakes can be made here, but on the whole, pre-mRNA splicing is what's actually occurring to get rid of those introns, which is amazing. But what are we going to do with it? Well, we're going to process this because we'll have some intermolecular cleavage. Those methylations can actually occur, and we're going to have a chemical conversion of nucleotides. But for us to actually do this, what we want to do is we want to think about what we're going to do with now that mature mRNA. And we use this, what's going to happen is this is a space-filling model of what's called TRNA, transfer RRNA. And what it does is it literally is inside the cytosol of your cell, and it uses a sequence down here. We call it the anticodon. And the anticodon will complementary base pair to the codon sequence on our now mature mRNA. And what it's doing on the far end is it's carrying that one amino acid. And way back when, chapters 1 through 5 or whatever it was, and I told you we will use dehydration synthesis reactions to bring these together. What's happening is you have temporary hydrogen bonding for the anticodon down here, but the actual dehydration synthesis reactions to form a peptide bond will be occurring up here. So that's some intricacies that we don't really need to worry about, the how and the why, except to say that in a few minutes I'm going to show you how does that anticodon know where to go, and why do you need to know that. So we have to decode the information in that mRNA, okay, to produce a polypeptide. And what's going to happen is we're going to catalyze all of these reaction groups using ribosomes, okay, because we're bringing the carboxy terminal group of one amino acid in an adjacent area to the amino group of another amino acid. And you'll see how that happens here. So inside your textbook, they have this figure. It should look fairly familiar to you. Here's one amino acid here. Here's another amino acid there. And look, there's a hydrogen and an OH. And using a dehydration synthesis reaction, we form that peptide bond. And that peptide bond is what holds now these two amino acids together. So we have had initiation, elongation, and termination. Okay? Well, how do I say this? I said that correctly. Let me correct myself here. Once we actually have processed our mRNA, we have a ribosome. We can now begin to initiate, elongate, and terminate. And what's going to happen is that mRNA is going to bind to a small subunit of the ribosome. We're going to find that AUG, okay? That's going to give us our methionine start site. And what's going to happen is tRNAs will move in, binding by those anticodon sequences, such that they will continue to process along until, as the chain moves into the endoplasmic reticulum, if that's where it's supposed to move to. And I'll show you a cartoon of how that would actually function. in lab, I send you off to look at a couple of different representations of movies of this, but the concept is pretty much the same. Here is our mature mRNA that has, all the structures in place it's supposed to have. And now we actually have our tRNA with the complementary base pair. Here's AUG inside our exon number 1. The complementary base pair is UAC, and that's carrying the methionine here. Now, a small subunit, mRNA, our second amino acid will come in along with our large ribosomal subunit. And we will continue this process. And what they're doing is, we're pointing out to you that you actually have 3 locations here: an ejection site, the principal site, and the acceptor site. And each time these two, P and A, become adjacent to one another, a dehydration synthesis reaction is going to add the next amino acid to this chain. And this continues. This shuffles along. It then enters into the ejection site, so the P would be ejected next. Okay, and that gets us to the end. So just to review, let's go up here, right? So this is the translation piece. The translation piece is pretty straightforward. Once we have processed our mRNA correctly, such that we have our primary RNA pre-mRNA transcript to be modified. And remember the modification, for as far as we're concerned, getting rid of those exons, throwing on a 5' cap, making sure a 5' untranslated region is there, 3' untranslated region, and a polyadenylation tail are present as well. So putting these 3 presentations together, we now can hopefully make sure we understand that if DNA is double-stranded and it has 6 possible reading frames, what's happening when we're just transcribing is we're figuring out where that ATG is going to be such that the correct RNA polymerase sits down using the transcription factors and the upstream regulatory elements that will then allow us to make a pre-mRNA, and that pre-mRNA will now be modified. And modification is going to make sure that it's stable till it can actually get to a ribosome where it will be translated. And at that translation, what we're going to do is we're going to make protein. But remember what I'm telling you here. First, we need our small subunit. We need our mature mRNA. So mRNA. Then we need the actual methionine met tRNA with tRNA, Jonathan. Okay? And then the large ribosomal subunit, or large. When you put all of these together, now you can actually begin to translate till we get to the stop codon, and then we get all the way back to the beginning of the semester when what's happened, of course, here is now we have our primary strand of amino acids. So we'll have a primary strand. Remember, those will fold on top of each other either using beta-pleated sheets or alpha helical structures, such that you end up making, if these are the secondary structures required for this to form, we end up forming now our correct tertiary structure, okay, being held in place by disulfide bridges and hydrogen bonding. And this is the, in fact, tertiary structure that makes it functional. Okay? So this is how you tie the sort of simplistic introduction that you got way back inside chapters 1 through 5 to now how we use molecular mechanisms for this to actually happen. Okay? Thank you very much.